Hi, I'm Dr. K. Yadav, Professor and Principal, Rajiv Gandhi College of Pharmacy, Bhopal, with another interesting and learning episode of B.Pharma on the key unit of Parenteral Products 5, which is a very important unit of the section of Pharmaceutical Manufacturing Technology 6th semester. Let us start our episode while taking a look at what are we going to learn today. There are 4 modules of all. Module 1, Aseptic Techniques, Sources of Contamination. Module 2, Methods of Prevention. Module 3, Design of Aseptic Area. In the last module, Module 4, Laminar Flow Bench Services and Maintenance. Module 1, Aseptic Techniques, Sources of Contamination. As this section is a part of Parenteral Products unit, let us briefly understand parenterals. Sterile products are dosage forms of therapeutic agents that are free of viable microorganisms. They include parenteral, ophthalmic, and irrigating preparations. Parenteral products are unique dosage forms because they are injected through the skin or mucous membranes into internal body compartments because they have circumvented the highly efficient first line of body defense, the skin and mucous membrane. They must be free from microbial contaminations and from toxic components, as well as possess an exceptionally high level of purity. Aseptic techniques are procedures carried out during the preparation of a sterile product in order to minimize contamination. Aseptic technique involves strict guidelines and its primary goal is to prevent the spread of microbial contamination. Let me just give you an example to justify the use of aseptic techniques. When a fungal meningitis outbreak swept across America in 2012, affecting over 800 people, the FDA traced its origin back to the compounding pharmacy, which was not using aseptic techniques in compounding sterile preparations. The incidences such as of 2012 meningitis outbreak underlines the importance of receiving proper training in aseptic techniques. Aseptic technique is a set of specific practices and procedures performed under carefully controlled conditions with the goal of minimizing contamination by pathogens. Aseptic technique can be applied in different settings, for example, in any clinical setting, operating room in a hospital, insertion of intravenous lines during compounding sterile preparations. However, in this module, we are focusing aseptic techniques specifically for parenteral preparations, manufacturing and handling. Before going on the next section, let us clearly understand some of the important terms used in aseptic processing. Asepsis. Asepsis is a state of being free from disease-causing contamination, such as pathogenic bacteria or preventing contact with microorganisms. Aseptic processing facility, a building or separate area containing clean rooms in which air supply, materials and equipment are regulated to control microbial and particle contamination. Critical area, an area designed to maintain sterility of sterile materials. Sterilized products, containers, closures and equipment may be exposed in the critical area. D-value. D-value is the time in minutes of exposure at a given temperature that causes a 1 log or 90% decrease in the population of a specific microorganism. HEPA filter, high efficiency particulate air filter, which has a minimum of 0.3 micron particle retaining efficiency of 99.97%. ULPA filter. ULPA is an acronym for Ultra Low Penetration Air filter, and ULPA filter can remove from the air at least 99.999% of dust, pollen, mold, bacteria and any airborne particles with a size of 100 nanometers or to say 0.1 micron or larger. Laminar flow, an airflow moving in a single direction and in parallel layers at constant velocity from the beginning to the end of a straight line vector. Sterile drug products produced by aseptic processing. Let us understand the basic difference between the production of a sterile drug product using aseptic processing and terminal sterilization. Terminal sterilization usually involves filling and sealing product containers under high quality environment conditions. Products are filled and sealed in this type of environment to minimize the microbial and particulate content of the in-process product and to help ensure that the subsequent sterilization process is successful. In most cases, the product, container and closure have low bioburden, but they are non-sterile. The product in its final container is then subjected to a sterilization process such as heat or irradiation. Whereas in an aseptic process, the drug product, container and closures are first subjected to sterilization methods separately as appropriate and then brought together, as there is no process to sterilize the product in its final container. It is critical that containers to be filled and sealed in an extremely high quality environment. Aseptic processing involves more variables than terminal sterilization. Before aseptic assembly into a final product, the individual parts of the final product are generally subjected to various sterilization processes. For example, glass containers are subjected to dry heat, rubber closures are subjected to moist heat, and liquid dosage forms are subjected to filtration. Sources of contamination. Contamination is a process or act that causes materials or surfaces to be soiled with contaminating substances. Both personal and material flow should be optimized to prevent unnecessary activities that could increase the potential for introducing contaminants to exposed product, container closures, or the surrounding environment. The person compounding is the most frequent cause of contamination, followed by contaminated equipment, contaminated raw material, and environmental contamination. Contamination by personnel. Proper attitudes of persons involved and strict adherence to aseptic techniques is essential to reduce the likelihood of contamination. Environmental contamination. Aseptic techniques requires control of the environment by maintaining proper airflow. HEPA provides a constant flow of air to the controlled area and the air is filtered. Laminar Airflow Workbench (LAFW) is designed to reduce the risk of airborne contamination during preparation of sterile products. Airflow control. Pre-filter removes the large particles, that is the lint, dust. High efficiency particulate air, the HEPA filters, removes 99.97% of particles and airborne microorganisms of 0.3 micron or larger. There are two types of laminar airflow workbench. The horizontal hood air flows horizontally across the work area, whereas the vertical air flows vertical downwards onto the surface of the work area. Such biologically safety cabinets are used in the preparation of anti-neoplastics and other potentially hazardous products. Some of the air is recirculated and filtered again. Remainder is vented to the outside. Such setting is safer for the preparer as the air is not blowing directly at them, avoiding the possibility of carrying potentially hazardous particles or small droplets. Coming to key elements of contamination. There are 4 areas of concern to get a better idea of the overall picture of contamination control. These are the things that need to be considered when providing an effective contamination control program. First is HEPA, the high efficiency particulate air filter. These filters are extremely important for maintaining contamination control. They filter particles, as I told you before, as small as 0.3 microns with an efficiency of 99.97%. The next is clean rooms. Clean rooms are designed to achieve and maintain an airflow in which essentially the entire body of the air within a confined area moves with uniform velocity along parallel flow lines. This airflow is called laminar flow. The more restriction of airflow, the more turbulence. Turbulence can cause particle movement. Third is filtration. In addition to the HEPA filter commonly used in the clean rooms, there are a number of other filtration mechanisms used to remove particles from gases and liquids. These filters are essential for providing effective contamination control. And the last is cleaning. Cleaning is an essential element of contamination control. Decisions need to be made about the details of clean room maintenance and cleaning. You can see on your screen how the pharmacists wear gloves and masks to ensure an aseptic environment. Module 2. Methods of prevention of contamination. Contamination can be prevented by using strict environmental controls. Most important is to have a clean room to perform all critical steps in which a sterile environment is maintained. Such rooms should have a restricted entry and only trained staff may be allowed to enter the room. Personnel working in such area must wear sterile clothing, avoid contact with non-sterile surfaces, practice proper hand hygiene, and know how to correctly handle equipment to minimize risk. Some of the important points which are useful for prevention of contamination are as follows. All items in a sterile field must be sterile. Sterile packages are opened as close as possible to time of actual use. There should be no talking, laughing, coughing or sneezing across a sterile field. Coming to the methods for prevention of contamination during aseptic processing. Operations shall be performed within specifically designed areas of adequate size. There shall be separate or defined areas or such other control systems for the firm. Operations as are necessary to prevent contamination or mix-ups during the course of the different procedures. Floors, walls and ceilings of the smooth, hard surfaces that are easily cleanable. Temperature and humidity controls should be maintained. An air supply filtered through high-efficiency particulate air filters under positive pressure regardless of whether flow is laminar or non-laminar. A system for monitoring environmental conditions. A system for cleaning and disinfecting the room and equipments to produce aseptic conditions. A system for maintaining any equipment used to control the aseptic conditions. Air filtration systems, including pre-filters and particulate matters as filters, shall be used when appropriate on air supplies to the production areas. Equipment shall be constructed so that the surfaces that contain components, in-process materials or drug products shall not be reactive, additive or absorptive so as to alter the safety, identity, strength, quality or purity of the drug product beyond the official or other established requirements. Equipment shall be cleaned, maintained and sanitized at appropriate intervals to prevent malfunctions or contamination that would alter the safety, identity, strength, quality or purity of the drug product beyond the official or other established requirements. Appropriate return procedures designed to prevent microbiological contamination of drug products purporting to be sterile shall be established and followed. Such procedures shall include validation of any sterilization process. Hope this episode is quite informative and you can have understanding of the basics of aseptic techniques, sources of contamination and methods to prevent contamination. The next module will give you more details on the designing of an aseptic area. Module 3. Design of aseptic area. The flow of components, drug product containers, closures, labeling, in-process materials and drug products through the building or buildings shall be designed to prevent contamination. Separate or defined areas of operation in an aseptic processing facility should be appropriately controlled to attain different degrees of air quality. depending on the nature of the operation. Design of a given area involves satisfying microbiological and particle criteria as defined by the equipment, components, and product exposed, as well as the operational activities conducted in the area. Clean area control parameters should be supported by the microbiological and particle data obtained during qualification studies. An adequate aseptic processing facility monitoring program also will assess conforms with specified clean area classifications under dynamic conditions on a routine basis. You can see in Table 1 the summary of clean area, air classifications, and the recommended limits of microbiological quality. As you can see in the Table 1, there are 4 types of clean areas starting from 100, 1000, 10,000 and 100,000. These areas are designated as per the particles of size 0.5 micron or greater per square feet. As per the microbial load, they are classified into 1, 7, 10 and 100. The ISO has designated them as ISO room 5, 6, 7 and 8. Aseptic processes are designed to minimize exposure of sterile articles to the potential contamination hazards of the manufacturing operations, limiting the duration of exposure of sterile product elements, providing the highest possible environmental control, optimizing process flow, and designing equipments to prevent entrainment of lower quality of air into the class 100, which is known as the ISO 5. Clean area are essential to achieve the high assurance of sterility. The layout of equipment should provide for ergonomics that optimize the comfort and movement of operators. The number of personnel in an aseptic processing room should be minimized. The flow of personnel should be designed to limit the frequency with which the entries and exits are made to and from an aseptic processing room, and the most significant, its critical area. The design of equipment used in the aseptic processing should limit the number of aseptic interventions by personnel. For example, personnel intervention can be reduced by integrating an online weight check device, thus eliminating a repeated manual activity within the critical area rather than performing an aseptic connection. Sterilizing the pre-assembled connection using the sterile in place, which is known as the SIP technology, also can eliminate a significant aseptic manipulation. Automation of other processes steps, including the use of technologies such as robotics, can further reduce the risk of the product. The sterile drug product and its container closures should be protected by equipment of suitable designs. Carefully designed curtains and rigid plastic shields are among the barriers that can be used in appropriate locations to achieve segregation of the aseptic processing line. Use of an isolator system further enhances product protection. Use of a double door or integrated sterilizer helps to ensure direct product flow, often from a lower to a higher classified area. Airlocks and interlocking doors will facilitate better control of the air balance throughout the aseptic processing facility. Airlocks should be installed between the aseptic manufacturing area, entrance, and the adjoining unclassified area. Clean rooms are normally designed as functional units with specific purposes. The materials of construction of clean rooms ensure ease of cleaning and sanitizing. Processing equipment and systems should be equipped with sanitary fittings and valves with rare exceptions. Drains are considered inappropriate for classified areas of the aseptic processing facility other than the class 100,000, which is an ISO 8 area. It is essential that any drain installed in an aseptic processing facility be of suitable design. Equipment should be appropriately designed to facilitate ease of sterilization. Equipment should not obstruct airflow, and in critical areas, its design should not disturb the unidirectional airflow. Two clean areas are of particular importance to the sterile drug product quality: the critical area and the supporting clean areas associated with it. The first one, the critical area, the class 100 ISO 5 area. A critical area is one in which the sterilized drug product, containers and closures are exposed to the environmental conditions that must be designed to maintain the product's sterility. Activities conducted in such areas, including the manipulations, for example, aseptic connections, sterile ingredient additions of sterile materials prior to and during filling and closing the operations. This area is critical because an exposed product is vulnerable to contamination and will not be subsequently sterilized in its immediate container. Air in the immediate proximity of the exposed sterilized containers and closures and filling and closing operations would be of appropriate particle quality when it has to be per cubic meter the particle count, which has to be not more than 3520 in a size range of 0.5 micron or larger. This level of air cleanliness is also known as the class 100 ISO 5. Secondly is the supporting clean areas. The nature of the activities conducted in a supporting clean area determines its classification. FDA recommends that the area immediately adjacent to the aseptic processing line meets at a minimum class of 10,000, which is ISO 7, standards under the dynamic conditions. Manufacturers can also classify this area as class 1000, the ISO 6, or maintain the entire aseptic filling room at class 100, the ISO 5. An area classified as a class 100,000, the ISO 8, air cleanliness level is appropriate for less critical activities, for example, the equipment cleaning. Finally, the clean area separation. An essential part of the contamination prevention is the adequate separation of areas of the operation. To maintain air quality, it is important to achieve a proper airflow from the areas of higher cleanliness to the adjacent less clean areas. It is vital for rooms of higher air cleanliness to have a substantial positive pressure differential relative to the adjacent rooms of lower air cleanliness. For example, a positive pressure differential of at least 10 to 15 pascals should be maintained between adjacent rooms of different classification where the doors are closed. When the doors are open, outward airflow should be sufficient to minimize the ingress of contamination, and it is critical that the time a door can remain ajar be strictly controlled. Before we move to our next module, let us recap what topics we have learned today. Till now, we have studied general introduction about the aseptic techniques, the purposes of aseptic techniques, sources of contamination, the key elements of contamination, control, and methods of prevention of contamination. Then we had information on design of aseptic area and clean room airflow principles. Now we will take a look at the laminar flow bench services and maintenance. Module 4, Laminar flow bench services and maintenance. Clean room airflow principles. Clean rooms maintain particulate-free air through the use of either HEPA or ULPA filters, employing laminar or turbulent airflow principles. Laminar or unidirectional airflow systems direct the filtered air downward in a constant stream. The laminar airflow systems are typically employed across 100% of the ceiling to maintain constant unidirectional flow. Laminar flow criteria is generally stated in a portable workstations, the LF hoods, and is mandated in an ISO 1 through ISO 4 classified clean rooms. Proper clean room design encompasses the entire air distribution system, including the provisions of adequate downstream air returns. In vertical flow rooms, this means the use of low wall air returns around the perimeter of the zone. In horizontal flow applications, it requires the use of air returns at the downstream boundary of the process. The use of ceiling mounted air returns is contradictory to proper clean room system design. The figure 2 shows the working of the horizontal laminar flow clean benches, and the figure 3 shows the working of a vertical laminar flow clean benches. In the horizontal laminar flow bench, the main important thing is the pre-filter and the blower, which you can see in the topmost part, which is shown in the right side of the figure 2, shown by point 1, 2 and 3. The first is pre-filter, second is blower, and third is the fluorescent lamp. Under that you will see there is the UV light, and under that you will see IV bar kit provision at 5. After that, the entire bench work, you will see the points 6, 7 and 8 show the front cover, 7 which has the 2 penetrations on the each sizes, and the ULPA filter, or it can also be the HEPA filter at point 8. The number 9 shows the tempered glass side panels. And finally, in the left hand side of the figure, you will see point number 10, 11 and 12, which shows the microprocessor control system at point number 10 on the topmost. The middle is the electrical outlet, and finally the bottom part is the stainless steel worktop. In difference to the figure 2, you will see that the figure 3 is the vertical laminar flow clean bench. In this, the main difference is of point number 1 and 2. Here, the point 2 is the fan which is placed on the right hand side of the equipment on the topmost part. Under that is the ULPA filter, which can also be replaced by the HEPA filter. The fluorescent lamp at point number 4. At 5 is the IV bar kit, and at point 6 is the airflow and the temperature sensor. Under that you will see that UV light provision is provided at point number 7. Point number 8 shows the tempered glass side panels, and the 2 penetrations for the service fixtures is shown in point number 9. Point number 10 shows the optical front cover, and the left hand side of the figure 3 shows the microprocessor's control system, the electrical output, and the stainless steel worktop. In horizontal flow models, the filtered air is then passed through the main chamber of the clean bench in a horizontal laminar unidirectional air stream and is exhausted through the front opening of the cabinet. Whereas in the vertical flow models, the filtered air is passed through the main chamber of the clean bench in a vertical laminar unidirectional air stream before being exhausted through the front opening of the clean bench. In horizontal laminar flow clean benches, there is a slightly reduced level of turbulence compared to the vertical flow clean benches due to the airflow not striking the work surface. However, vertical laminar flow clean benches generate less turbulence around large pieces of equipment as compared to the horizontal laminar flow clean benches. Laminar flow clean benches. The main body of the clean bench is constructed of industrial grade electro-galvanized steel. One piece formed stainless steel work surface with a curved front. The edge is designed for maximum operator comfort. Lighting of 5000 K provides excellent illumination of the work zone and reduces operator fatigue. Laminar flow clean benches have been tested for cross-contamination and product protection using microbiological test methods. Each clean bench is individually factory tested for safety and performance in accordance with international standards. High performance blower. The system's centrifugal motor blowers with external rotor designs. The motors selected for the energy efficient, compact design and flat profile. It completely integrates the assembly, optimizes the motor cooling. All of the rotating parts are balanced for smooth, quiet and vibrating free operation. The blower system maintains the airflow as the filter becomes loaded, ensuring an optimum efficiency and product protection. An additional disposable pre-filter traps the large particles in an inflow air prior to reaching the main filter. This protects it against the damages and prolongs the filter life. ULPA filtration system. ULPA filters typically have an efficiency of more than 99.999% for a size of 0.1 to 0.3 micron filters and are superior to the HEPA filters, as the HEPA filters have an efficiency of 99.97%. The ULPA filters last as long as the conventional HEPA filters and have similar replacement costs. That the laminar flow clean benches provides the ISO class 3 to class 5 air cleanliness within the work zone as per the ISO on clean benches. User interface. An angled front, curved work surfaces, front edges and glass slides promotes the ergonomics. Just to inform you, ergonomics is associated with the way the worker is working in the workbench. Next is microprocessor control system. The soft touch controls for blowers, light, outlet and UV are easy to clean. A temperature compensated air velocity sensor and real-time displays allows the airflow to be monitored more accurately compared with the conventional pressure gauges. Summary. In summary, aseptic techniques are procedures carried out during the preparation of a sterile product in order to minimize contamination during manufacturing. With all these information, here we come to an end of today's lecture. Do keep in mind what we have discussed today. Always keep time for self-study. This is Dr. Khush Yadav signing off. If you want to learn more and enhance your knowledge, you may log on to our website www.cc.nic.in for MCQs, quizzes and LORs. Till then, keep studying. Goodbye.